Okay, so we are going to look at still on urine. Okay, urine formation. Okay, the last lecture we stopped up to this slide. Yes. Okay. So basically, when you are looking at this picture, okay, this diagram, you should be able to explain on the structure or the structure of kidney, the nephrons, okay, the functional unit of kidney, which is nephron, that involves in urine formation. Okay, so you should know the component of the nephrons, okay, where it it consists of blood vessels, starting with the glomerulus, which receives blood from the afferent arteriole, and the blood from the afferent arteriole enters into the glomerulus and causes the blood to flow with high pressure, and the blood will flow out of the glomerulus through the efferent arteriole. Okay, so this is the efferent arteriole. So the diameter of afferent and efferent arteriole are different, where the afferent arteriole will have a bigger diameter, and efferent arteriole will have a smaller diameter, and causes the formation of high blood pressure in the glomerulus, and causes blood fluid in the blood plasma, to enter into the Bowman capsule. Okay, so the Bowman capsule is the tubule part of the nephron. Okay, so if once the fluid from the blood or the blood plasma enters into the Bowman capsule, we call it as filtrate. Okay, itu kena faham lah. Okay, so fluid sepanjang-panjang yang selalu warna kuning ni, kita akan namakan dia filtrate. Okay, so after the Bowman capsule, you have the proximal tubule, where reabsorption of nutrients occurs. Okay, you have the reabsorption of water, nutrients such as amino acids, such as glucose. Okay, that will be reabsorbed back into the blood vessels. Okay, so the blood vessels surrounding the proximal tubule and also the distal tubule, in the cortex region of the kidney, we call it as peritubular capillaries. Okay, so this substance, okay, these molecules will be reabsorbed back into the blood, right? And then after that, this process we call it as reabsorption. Okay, reabsorption. Okay, so this is the reabsorption process, and then hydrogen ion will be secreted into the filtrate, and the transport epithelium of the proximal tubule will produce and secrete ammonia. Okay, so this ammonia will combine or will bind to hydrogen ion in the filtrate, forming ammonium ion. So this is to make sure that your blood maintains at the right pH, which is pH 7.4. Okay, so that is one mechanism of maintaining the blood, your body fluid at the pH of 7.4. Salah satu cara. Nanti bila kita tengok chapter respiration, ada lagi cara lain untuk maintain blood pH at 7.4. Bila kita tengok chapter circulatory system pun, kita akan tengok ada lagi satu mekanisme yang akan yang your body will undergo, okay, to make sure that your blood is maintained at 7.4. Okay, so and then after that, the filtrate will go down the loop of Henle, okay, and then in the descending limb of loop of Henle hanya akan berlaku reabsorption of water, because in the descending limb of loop of Henle there will be many aquaporins. Okay, so the presence of this aquaporin will help in the reabsorption of water. Okay, so and then after water has diffused out, so at the elbow of the loop of Henle, there will be the filtrate concentration will be very concentrated, which is 1200 milliosmol. So due to the high concentration of filtrate in the elbow of loop of Henle, so when the filtrate goes up, okay, goes up through the ascending limb of loop of Henle, so for the ascending, it has two segments. Okay, first you have the thin segment, second you have the thick segment. Okay, so in the thin segment, sodium chloride will diffuse. Okay, the process is diffuse, facilitated diffusion lah, that is. Okay, so diffuse out of the thin segment of ascending limb of loop of Henle. Sebab apa? Sebab concentration filtrate dekat elbow ni dah terlalu tinggi, so sodium chloride boleh keluar macam tu je. So but as the filtrate goes up in the thick segment, the process still wants sodium chloride to move out of the filtrate, but the process involved is through active transport. Okay, so and then as the filtrate moves up, it enters into the distal tubule, and then in the distal tubule you have the process of reabsorption, where bicarbonate ion will be reabsorbed, water will be reabsorbed, and also sodium chloride also will be reabsorbed. Potassium ion, hydrogen ion will be secreted into the filtrate, and then the filtrate, because sodium chloride moves out, okay, you have bicarbonate ion moves out. At this part, at this tubule, the concentration of filtrate will be dilute. Okay, will be dilute. Okay, but as the filtrate moves down the collecting ducts, okay, you'll see that sodium chloride will start to move out also by through active transport. So as sodium chloride moves out, as you can see, the concentration of filtrate will be concentrated also due to the movement or reabsorption of water and also reabsorption of urea. Okay, so by looking at this diagram, you should be able to know the formation of urine. Okay, the process that involves kat mana berlaku ultrafiltration, kat mana berlaku reabsorption, kat mana berlaku secretion. One more thing, okay, because you observe that water diffuse out in the collecting ducts, so in the collecting ducts you also have plenty of aquaporins. Okay, aquaporins helps in the reabsorption of water. Okay, so alright, so now we are going to look at, tadi kita recap apa yang kita dah belajar last week. So now we are going to look at the next part, which is solute gradient and water conservation. Alright, so it says here urine is much more concentrated than blood. So this is the concentration of urine, which is 1200 milliosmol per liter, which is in the blood, the concentration of blood is about 300 milliosmol. Okay, so bandingkan, so your urine will be very concentrated. So and then you have the juxtamedullary nephrons. Juxtamedullary nephrons, where the loop of Henle extend very deep. It has a long loop of Henle. Okay, so extend deep into the medulla, which are essential for the production of urine that is hyperosmotic to body fluid. Okay, so saya pernah baca dalam buku rujukan Campbell, tak silap, dia kata ini adalah adaptation untuk haiwan yang duduk kat tempat yang panas. Okay, bila dia duduk tempat yang panas, dia nak conserve water in the body. Okay, the animal wants to conserve water in the body, so they will have plenty of juxtamedullary nephrons. Okay, so that reabsorption of water will occur dengan banyaknya, berbanding kalau animal tu dia ada cortical nephron saja, kan? So cortical nephron will have a very short loop of Henle, so not much water will be reabsorbed. So this is an adaptation of animal that lives in hot area to conserve water. So this animal will produce urine in with low volume and very concentrated. Okay, hyperosmotic. Okay, very concentrated. So next, Okay, so the cooperative action and precise arrangement of the loops of Henle and collecting ducts are largely responsible for the osmotic gradient that concentrates the urine. Okay, that concentrate the urine. So kedua-dua ni, loop of Henle and collecting ducts, plays a very major role in the reabsorption of water and also reabsorption of sodium chloride and another solute, which is urea. Okay, so the reabsorption of urea which will occur only at the collecting ducts. Okay, kejap ya kelas. Okay, so sorry. Alright. So okay, so responsible for the osmotic gradient that concentrate the urine. So you have to know that in the formation of urine ada dua concentration gradient yang awak kena take note. Satu, concentration of filtrate in the tubules and also concentration of interstitial fluid, okay, surrounding the medulla, okay, medulla region. Okay, so sodium chloride and urea contribute to the osmolarity of the interstitial fluid. Saya short form as IF. So which causes reabsorption of water in the kidney and concentrate the urine. Okay. Okay. Alright. Okay, so concentrating urine in the mammalian kidney using the two solute model. Okay, so when you see this model, it says two solute, it means that it involves sodium chloride, okay, and also urea. Okay, and also urea. Alright. So it says here in the proximal tubule, filtrate volume decreases but its osmolarity remains the same. You boleh dengar saya lagi ke?
Boleh, madam.
Okay, sorry saya macam terkeluar tadi pada yang okay, so in the proximal tubule, filtrate volume decreases. Okay, filtrate volume decreases, but its osmolarity remains the same. So filtrate passing from the Bowman capsule to the proximal tubule has osmolarity of about 300 milliosmol, which is the same as the blood. Large amount of water and salt is reabsorbed from the filtrate as it flows through the proximal tubule in the renal cortex. Kenapa awak agak Bowman capsule ni, dia punya concentration dia sama dengan blood? Sebab apa? Tadi kita observe in the glomerulus, due to the high pressure, many nutrients, okay, including sodium chloride and also water enters into the Bowman capsule. Okay, so that's why the osmolarity of the blood, okay, in the cortex region and also in the Bowman capsule is fairly similar, fairly the same, which is 300 milliosmol, which is the same as the blood. Okay, so a large amount of water and salt is reabsorbed from the filtrate as it flows through the proximal tubule in the renal cortex. So kita tengok ini adalah apa yang akan berlaku pada cortex region. Kan kita remember tadi cortex region ada glomerulus and also Bowman capsule and also proximal tubule. Okay, okay, so as the filtrate flows from the cortex to the medulla, bahagian bawah kan, so bahagian bawah ataupun the middle region of the kidney Okay, the medulla is separated into two regions, which is the outer medulla and also the inner medulla. Okay, inner medulla yang paling dalam lah. Okay, so to the medulla, in the descending loop of Henle, water leaves the tubule by osmosis. Okay, osmosis. So you also have presence of aquaporins. Okay, aquaporins in the wall of descending loop of Henle. So solute including sodium chloride becomes more concentrated, increasing the osmolarity of the filtrate. So bila filtrate tu turun ke bawah, kan, turun descending limb of Henle, so air keluar. So nanti bila filtrate reach at the elbow of loop of Henle in the inner medulla region, okay, so the filtrate concentration will be very concentrated, okay, increasing the osmolarity of the filtrate, which is 1200 milliosmol. Okay, so the highest osmolarity in filtrate occurs at the elbow of loop of Henle. This maximizes the diffusion of salt out of the tubule as the filtrate rounds the curve and enters the ascending limb, which is permeable to salt but not to water. So bila filtrate tu dah masuk dekat ascending, dekat bahagian thin segment, dia akan jumpa thin dulu baru thick. Okay, so sodium chloride will diffuse out, but the process is not it is passive, but it is through facilitated diffusion, because in order for sodium chloride to move out of the filtrate into the interstitial fluid, it has to use protein channel. Okay, transport protein, alright. So sebab apa? Sebab molekul ni adalah molekul yang bercharge, kan. Kita dah belajar sifat plasma membrane, dia tak boleh allow ion to just simply diffuse out. So the ion will have to use transport protein. So the process is through facilitated diffusion. So which is permeable to salt but not water. So in the ascending, takkan berlaku, there will be no reabsorption of water, no movement of water whatsoever. Okay, in the ascending limb. So this help maintain a high osmolarity in the interstitial fluid of the renal medulla. So kita ada region tadi, kan. Kita ada cortex, kita ada medulla, outer medulla dengan inner medulla. Sebab apa? Sodium chloride keep on moving out, okay, in the inner medulla and also in the outer medulla region. So this cause the interstitial fluid osmolarity to increase. Okay, to increase. Sebab apa? Adanya pergerakan sodium chloride keluar daripada filtrate kepada interstitial fluid. Alright, so loop of Henle has several qualities of countercurrent system, which involves the loop to expend energy to actively transport sodium chloride. Okay, expend energy to actively transport sodium chloride occurs where? Occurs in the thick segment. Okay, in the thick segment. So in order for sodium chloride to move out of the filtrate in the thick part of the loop of Henle, it uses energy. Okay, active transport. So from the filtrate in the upper part of the ascending limb of loop of Henle, okay, which is in the thick segment lah. So this maintain a high salt concentration in the interior of the kidney, enabling kidney to form concentrated urine. Okay, kenapa perlunya? Why? Okay, is it really essential to make sure that interstitial fluid osmolarity is maintained at a high concentration? Okay, so sebab apa? Kita kena tengoklah dekat descending limb of loop of Henle, air keluar, kan. So macam mana air nak keluar? Tadi proses dia cakap through osmosis, kan. Osmosis adalah proses passive transport. Okay, pergerakan air daripada high water concentration to low water concentration, kan. So kat luar ni will be very concentrated. Okay, will be very concentrated. So that's why water can always diffuse out of the descending limb of loop of Henle, disebabkan interstitial fluid concentration is high, disebabkan apa? Sodium chloride selalu akan keluar dekat ascending. So dia akan saling melengkapi proses tu. Okay. Okay, so countercurrent system that use energy to create concentration gradient is called countercurrent multiplier system. Countercurrent multiplier system. Multiplier system, why? Because it uses energy to actively transport sodium chloride to cause interstitial fluid to be very concentrated. Countercurrent because it involves fluid to flow in the opposite direction. Okay, fluid. Kalau tengok descending, filtrate moving down, ascending, filtrate moving up. Okay, so countercurrent. Countercurrent, apart from it involves movement of fluid in opposite direction, it also involves the exchange, okay, exchange of materials. Okay, materials. In this case, the exchange of water and also sodium chloride. Okay. So the filtrate is hypoosmotic to body fluid by the time it reaches the distal tubule. Lepas ascending, kan, lepas thick segment, baru dia akan jumpa distal tubule. Okay, so sodium chloride keluar. Bila filtrate reaches reaches distal tubule, the concentration or the osmolarity of filtrate, okay, will be low. Okay, will be low. Sebab sodium chloride dah keluar tadi kan. Okay, so this causes the filtrate to be hypoosmotic, by very dilute. Okay, dilute. Okay. Okay, so vasa recta. Okay, vasa recta, blood capillaries that surround loop of Henle of juxtamedullary nephron. So blood vessels surrounding loop of Henle, we call it as vasa recta. Blood vessels surrounding the tubule at the upper region, at the cortex region, surrounding the proximal tubule and distal tubule, we call it as peritubular capillaries. So you should know the name of these blood vessels. Okay, so the vasa recta will surround the loop of Henle, so divided into descending and ascending vessels of vasa recta. So vasa recta, dia juga ada descending dan dia juga ada ascending vessels. Okay, so that carry blood in opposite direction through the kidney's osmolarity gradient. Opposite direction to what? Opposite direction to the filtrate. Okay, in the loop of Henle, in the ascending and descending. So if you are referring to this diagram, it is a bit confusing. Saya nak awak elakkan rujuk gambar ni sebab dia macam confuse sikit, saya tak suka guna. It is better for you to use this diagram to refer to. Okay, so vasa recta. So as you can see, that's why the process tadi kita namakan dia juga sebagai counter current multiplier system. Okay, multiplier sebab apa? Dia guna energy untuk sodium chloride move out of the filtrate kan, dekat thick segment. Okay, counter current tadi kita tengok aliran filtrate is in opposite direction in the descending and also ascending, and also to the blood flow in the vasa recta. Okay, blood flow in vasa recta will be in opposite direction. Okay, opposite direction to the loop of Henle. So kalau if we are looking at descending limb, okay, descending filtrate bergerak ke bawah kan. Okay, so the blood vessels, the ascending vessel will surround the descending limb of loop of Henle. Nampak ya, saya ulang. Descending loop of Henle turun, blood vessels, okay, kita namakan dia sebagai ascending vasa recta, ascending vessel. So pergerakan darah tu adalah in opposite direction of the blood. Of the filtrate in the descending. So pergerakan darah dengan filtrate yang mengalir sebelah-sebelah tu is in opposite direction. Sama juga bila kita tengok kepada ascending, okay, ascending loop of Henle. So filtrate naik atas. Okay, kalau blood vessels yang warna merah ni kita panggil dia sebagai descending vessels of vasa recta. So darah akan flow in opposite direction, which is dia akan bergerak ke bawah towards the inner medulla region. Okay, the inner inner medulla region. So that's why process tu is countercurrent multiplier system. The flow of the blood in the vasa recta will flow in the opposite direction as filtrate in the loop of Henle. Okay. So when this fluid flows in the opposite direction, there will be exchange of materials, which is water and also sodium chloride. Alright. Okay. So ini kita dah go through jap. Balik slide tadi. Okay, so what prevents the capillary of the vasa recta from dissipating the gradient by carrying away the high concentration of sodium chloride in the medulla's interstitial fluid? Okay, kan kita observe tadi sodium chloride ni keep on moving out of the ascending kan. So bila dia keep on moving out of the ascending, proses tu akan membantu, okay, sekejap, prevent capillaries from okay. Okay, ascending, okay, ascending limb, fokus kepada ascending limb of loop of Henle, sodium chloride keluar. Bila dia sebelah dengan descending vessels, okay, of vasa recta, darah mengalir turun ke bawah kan. So sodium chloride akan ada tendensi untuk masuk dalam darah, dalam descending vessels, okay, descending vessels. So macam mana proses ni nak memastikan interstitial fluid ni will always have a high concentration of solute bila sodium chloride yang keluar daripada filtrate masuk dalam distal tubule, masuk dalam descending vessels. So ada proses dia yang terlibat. Okay, so kita akan tengok satu-satu lah. It says here, as the descending vessels conveys blood towards the inner medulla, water is lost from the blood. Okay, so sodium chloride is gained by diffusion. Gain ni awak take note So maksudnya pergerakan tu daripada filtrate dalam ascending limb of loop of Henle, daripada filtrate keluar ke interstitial fluid sekejap, lepas tu masuk balik dalam blood, which is in the descending vessels of vasa recta. Okay. So is gain. Gain tu maksudnya masuk dalam darah, dalam descending vessels of vasa recta. Okay, fluid, filtrate, and blood will flow in the opposite direction. Okay, so okay. Tak apalah, nanti saya draw balik semula. Okay, kita tengok slide ni dulu. So net fluxes are reversed as blood flows back towards the cortex in the ascending vessels, with water re-entering the blood and sodium chloride diffuse out. Okay, so vasa recta are able to supply the kidney with nutrients without interfering with the osmolarity gradient in the inner and outer medulla. Okay, so concentrating urine in the mammalian kidney, the two solute model. The countercurrent-like characteristics of the loop of Henle and the vasa recta help to generate the steep osmotic gradient between the medulla and the cortex. Okay. So, okay, ada 3.5. Alright. So, ini saya okay. So, sekarang ni kita nak fahamkan konsep countercurrent multiplier system. Okay, system. Tu solute model juga terlibat, dia seiring kan, involving the two model. Okay, tu solute. Sekarang ni satu solute je kita tengok kan, baru, which is sodium chloride. Urea kejap lagi kan, kejap lagi dia punya contribution dia. Okay, so saya akan guna yang warna orange ni, yang warna slight orange, yellowish color ni as the tubule. Okay. So ini adalah of loop of Henle. Okay, descending limb of loop of Henle. So filtrate turun. Okay, filtrate turun. Okay, so sebelah dia kita ada saya buat warna biru lah. Okay. Biru adalah warna untuk blood vessels that carry deoxygenated blood. Okay, in this case, blood will flow in the opposite direction, and this is the ascending vessel of vasa recta. Okay, so tengok tu region yang terlibat, kita ada beberapa region, tapi kita nak concentrate kepada 2 region sahaja kat sini. Okay, kita ada yang atas is the region outer medulla. Okay, bahagian bawah is the region inner medulla. Okay, so sebelah descending limb kita ada apa? Sebelah descending limb kita ada apa? Sebelah descending limb of loop of Henle, we have what? Oh, saya terkeluar ke tadi? Saya tak present slide ke apa? Am I not presenting any slide? Present. Ha. Okay, so sebelah descending limb of loop of Henle, we have what?
Descending limb.
Ha. Thank you, ya. Yes, see. Ascending limb of loop of Henle. Okay, so blood vessels yang sebelah dia. Sekejap. Ascending darah. Filtrate naik ke atas kan. So, saya draw dia balik semula. Saya draw lah. Ini. Okay, ini thin segment. Okay, thin. Thin. Ini adalah thick. Okay. Okay, so the descending limb will flow filtrate upward. Okay, towards the cortex that enters into the distal tubule. Okay, sebelah distal tubule, we will have another blood vessels that flows blood in the opposite direction. Okay, from cortex into outer medulla into the inner medulla region. So this is the descending vessels of vasa recta. Okay, so countercurrent multiplier system, multiplier due to sodium chloride that moves out of the thick segment using energy. Sodium chloride keluar using energy. Okay, countercurrent due to the flow of filtrate and blood which is in the opposite direction, and it involves exchange of materials which is water and also sodium chloride. Kalau kita perhatikan dekat sini, okay, sodium chloride keluar kan, dia akan keluar into interstitial fluid. Okay, into interstitial fluid, dia akan masuk dalam darah, dalam descending vasa recta kan, dia akan masuk dalam descending vasa recta. So descending vasa recta dia akan naik semula kan, dia akan berjumpa semula. Kan, dia akan berjumpa semula dengan dengan ascending. Kan, dengan ascending. So, sodium chloride yang dah masuk dalam darah ni, kan, sodium chloride yang dah masuk dalam darah, dia masuk dalam darah, dia masuk dalam darah. Okay. Okay, so awak kena fahamkan yang keadaan yang dekat descending limb of loop of Henle. Sodium chloride moves out into interstitial fluid, into blood in the descending vessels. Okay, descending vessels of vasa recta. So, bila darah, dia jumpa dengan aliran filtrate dekat descending limb ni. Okay, descending apa yang berlaku? Air keluar kan? Okay, water diffuse out. Sorry, H2O. H2O. Okay. So so the water in the filtrate that moves into the interstitial fluid akan masuk dalam filtrate. Sorry, akan masuk dalam blood. Okay, akan masuk dalam blood. Bila blood air masuk, sodium chloride akan keluar balik semula. Okay, sodium chloride akan keluar balik semula. Okay, sebab air masuk. Air masuk dalam darah, dalam ascending vessels. Same goes to what happened in the descending vessels. Bila sodium chloride masuk dalam darah, yang akan keluar daripada darah adalah water. Okay, itu cara dia nak balance fluid concentration, especially in the interstitial fluid. So, kita check balik semula. Okay, dia cakap apa kat sini? Net fluxes are reversed as blood flows back towards the cortex in the descending vessels, which water re-enter the blood. Okay, water re-enter the blood and sodium chloride diffuse out. Dekat sini lah kan. Okay, dekat sini darah mengalir ke atas. Dia cakap apa? Air masuk balik ke dalam darah. Okay, air daripada descending masuk balik ke dalam darah dalam ascending vessels. Okay, this air will be conserved in your body lah kan. So, it will be conserved in your body. The process of reabsorption tu. Okay, itu significant reabsorption of water. Filtrate, keluarkan air, masuk ke dalam ascending vessels, so that your blood will reabsorb back water and the water will circulate in your body. Kan, so dia menghalang air ni keluar menjadi urine. Okay. Okay, so vasa recta are able to supply the kidney with nutrients without interfering with the osmolarity gradient in the inner and outer medulla. Okay, darah yang dalam blood vessels warna merah ni, dia adalah oxygenated blood. So, bila dia adalah oxygenated blood, it also brings nutrients. Okay, nutrients. Bila sodium chloride keluar, dia juga keluar bersama dengan nutrients untuk supply cells of the kidney with nutrients. Okay, So bila awak baca awak punya nota ni, awak kena refer kepada diagram juga. Kan, bila awak refer kepada diagram, it will be much more clearer for you to understand. Okay. Okay, so the countercurrent characteristics of the loop of Henle and the vasa recta help to generate a steep osmotic gradient between the medulla and the cortex. Okay, so nutrient keluar, sodium chloride juga keluar due to here kan. So this causes the interstitial fluid concentration will be high kan. So the high concentration of interstitial fluid will contributes to the reabsorption of water in the descending limb of loop of Henle. Okay. So, bila kita ada region ni, kat atas ni kan kita ada cortex. Okay, from the cortex moving to the outer medulla, moving into the inner medulla, concentration of interstitial fluid akan menjadi lebih pekat. Okay, lebih pekat sebab nak memastikan air sentiasa reabsorb dekat descending limb of loop of Henle and also juga dekat collecting ducts. Okay. Okay, so now kita akan moving on to the next part, kan? InsyaAllah. Okay. So from distal tubule, filtrate descends towards the medulla. This time in the collecting ducts. Okay. Daripada distal tubule tadi kita tengok kan, dia filtrate daripada descending limb of loop Henle, it enters into the distal tubule, and then from the distal tubule into the collecting ducts. Collecting duct filtrate turun ke arah inner medulla. So the collecting ducts conducts the filtrate towards the osmolarity gradient. Maksudnya interstitial fluid getting the osmolarity of interstitial fluid will be getting higher. Okay. So as the filtrate passes from the cortex to the medulla, it encounters interstitial fluid of increasing osmolarity, kan? So awak kena take note, daripada cortex to the medulla, interstitial fluid concentration or osmolarity will increase. Okay, increase due to sodium chloride yang keluar. Okay, yang keluar daripada ascending loop of Henle. Okay, ascending limb of loop of Henle. So this result in more water exit the filtrate by osmosis and concentrating salt and urea and other solute in the filtrate. Okay, so this one you have to take note. We are concentrating on the filtrate movement at the collecting ducts. Okay, so bila filtrate turun collecting ducts, okay, air akan keluar. Bila air keluar, concentration filtrate akan meningkat. Akan meningkat. So bila dia meningkat, dia akan juga menyebabkan salt and urea, okay, to move out of the filtrate into interstitial fluid. Okay, into interstitial fluid. So urea diffuses out of the collecting duct as it traverses, okay, the inner medulla and contributes to the high interstitial osmolarity of the inner medulla. So you have to take note, there are two solutes which contributes to the interstitial fluid osmolarity getting higher as the filtrate moves from outer medulla into the inner medulla due to this solute, sodium chloride and urea. Urea hanya akan keluar daripada filtrate dekat collecting ducts in the inner medulla region. Okay, in the inner medulla region. Okay, so form the osmotic gradient that enables the kidney to produce urine that is hyperosmotic to the blood. So, as filtrate loses salt and urea, okay, salt and urea and also water is reabsorbed, this produces hyperosmotic urine. Okay, hyperosmotic urine. Alright. So, if you look at this diagram here, alright. Okay, so it gives you the concentration of filtrate. Okay, it shows you the concentration of filtrate in each region of the kidney. So we have the cortex, we have the outer medulla, and then we have the inner medulla. Okay, inner medulla. So as the concentration of filtrate, okay, the concentration of interstitial fluid daripada cortex, outer medulla and inner medulla will the osmolarity will increase. Okay, increase due to the movement of these two solute, sodium chloride and also urea. Pergerakan urea hanya akan berlaku dekat collecting ducts in the inner medulla region. Okay, in the inner medulla region. Air akan keluar daripada filtrate dekat descending limb of loop of Henle. Dia akan masuk ke mana? Ke ascending vessels of vasa recta. Okay. So dia air akan reabsorb masuk balik dalam blood circulation awak. Okay. So kalau dekat ascending limb of loop of Henle akan berlaku apa? Sodium chloride diffuse out and then actively move out of the filtrate. Okay. So this contributes to the high osmolarity of interstitial fluid. Okay, in the medulla region. In the inner medulla region, dia lagi tinggi sebab apa? Apart from sodium chloride, dia tu siapa? Interstitial fluid lah. Interstitial fluid juga akan terima urea yang keluar daripada filtrate in the collecting duct region. Okay, So you have to know what happened in each region. So kalau awak observe filtrate concentration dekat cortex region will be similar. Okay, will be similar in the proximal tubule will be similar to blood. Tapi bila dekat distal tubule, the concentration of filtrate will be very dilute, which is 100. Sebab apa? Due to sodium chloride yang asyik-asyik keluar daripada ascending limb of loop of Henle. So concentration of fluid will be very dilute, banyak air lah dekat filtrate ni. Okay, so but as the filtrate moves down, due to water that will be reabsorbed also. Okay, concentration of filtrate akan meningkat sebab air juga akan reabsorb dekat collecting ducts. Okay, and also because filtrate concentration getting higher as it moves down into the inner medulla, sodium chloride and also urea will also move out. Okay. So